Ever pondered what prompts that familiar rumble in your stomach, signaling it's time for a meal? The answer may lie within a cluster of cells nestled near the underside of the brain named AgRP neurons. These cellular heroes have a front-row seat to the body's energy levels, receiving hormonal updates from the stomach and fat tissue via the brain's blood supply. When energy dips low, these neurons spring to action, interacting with various brain areas to promote feeding. Scientists have uncovered some intriguing insights about these neurons, suggesting they might be the key to the mystery of hunger. Switching off these neurons in adult mice leads to a startling result. The mice cease to eat, potentially to the point of starvation. On the flip side, activating these neurons sends mice into a feeding frenzy, gorging themselves on available food. An intriguing experiment conducted by a team led by neuroscientist Amber Al-Hadef further revealed the adaptive nature of these neurons. Mice were treated to a calorie-free snack, a gel sweetened artificially. As the mice consumed the gel, the activity of the AgRP neurons dipped, as anticipated. However, this was a temporary response. The mice soon learned the snack was devoid of nutrients, and with each bite the neurons reacted less, fine-tuning the hunger response based on actual nourishment. The team also began deciphering the messages sent from the stomach to these neurons, revealing that the type of nutrient plays a role. Fat in the gut sends a signal via the vagus nerve, which stretches from the digestive tract to the brain. Meanwhile, glucose, a simple sugar, communicates with the brain through nerves in the spinal cord. The role of AgRP neurons in conditions such as anorexia has also been explored. Researcher Sutton Hickey closely monitored the eating habits of mice with activity-based anorexia, or ABA. She discovered these mice ate fewer meals, and even when they did eat, their AgRP neuron activity didn't decrease as it should have post-meal. This suggested a malfunction in the way the neurons responded to hunger and food cues. However, Sutton Hickey also found a potential solution. By genetically modifying the ABA mice so that their AgRP neurons were activated when a particular chemical was injected, these mice ate more meals and put on weight. This highlights the critical role these neurons play in regulating hunger and feeding behavior. The next step is to understand why AgRP neurons respond abnormally in ABA mice. Sutton Hickey hopes to identify a key molecule that could be targeted with a drug, potentially offering a new treatment avenue for conditions like anorexia and overeating. This ongoing research underscores the fascinating complexity of our bodies and the potential held within our cells to control and influence our behavior.